Hi everyone, I'm Sarah. Today I will talk about Diel variation in the upper layer biophysical process using a BGC-Argo in the Bay of Bengal. The ocean is the important ecosystem for biological productivity. Phytoplankton are the main primary producer in the ocean. Phytoplankton growth depends on sunlight. The solar radiation change during the day we call Diel variation. And the sunlight change can cause the phytoplankton different response, such as photoacclimation and vertical migration. I will talk later. In previous study in the Bay of Bengal show physical and biological factors change not only daily but also seasonally. However, past data too sparse to capture daily change. So in the study, we use high-frequency BGC-Argo float data to study the Diel variation. And this picture, we can see many colorful dots, and one dot represents one observation, and the different color indicate different sample frequency. And we use the high-frequency float, and is the red dot. So our float is location is here. And the BGC-Argo float is the part of the global ocean observation system. With that float, it can measure every 5 hours and measure temperature, salinity, chlorophyll, and dissolve oxygen. We use the date from August 24 to September 23, 2015. And we use the data above 80 meters for analysis. When we get the data, we need to remove long-term change to extract the Diel variation from the data. And we also use the sphere drop model to check if light limits phytoplankton growth. In the results, we can look at the float trajectory. The float moves through anti-clockwise AD and later anti-clockwise, cyclonic AD. When the cyclonic AD, the upwelling happens. So we can look at the picture. It has the two lights. The black is the mixed layer depth. The blue is isothermal layer depth. Is the depth where temperature becomes 0.5 degrees lower than the surface. And here is the upwelling occur. So it brings deeper water to the surface. So the isothermal layer depth is become shallower. Okay. Therefore, the upwelling is one of the long-term signals. So the original observation contains both long-term and Diel variation. To focus on the daily change, we use the anomaly analysis to remove long-term change. So this is our results. The Y is anomaly. Its formula is like here. Now we are looking at the Diel variation of biophysical parameter. First is temperature. Temperature increase during the day and peak in the afternoon because the solar heating. And dissolved oxygen. It's similar to temperature. It increases during the day as photosynthesis produces more oxygen. Salinity and mixed layer depth, they have no significant Diel variation. Diel variation. And now we look at the chlorophyll. And we want to know why surface chlorophyll decrease around the noon. So we use the two picture is chlorophyll at 10-meter depth, and the mean chlorophyll is average 0 to 80 meters. Mean chlorophyll is increased throughout the day and peak at 18. However, surface chlorophyll continue decrease until 11 o'clock. So if the chlorophyll decrease, mean phytoplankton should decrease, right? But it's not. The mean chlorophyll is rising. So it's two reasons. First is photoacclimation. At noon, sunlight is very strong, so phytoplankton reduce their chlorophyll to protect themselves. And another is vertical migration. Phytoplankton move deeper to avoid strong light and obtain more nutrients. So it's not mean lower chlorophyll means lower phytoplankton. So we want to know whether light limits phytoplankton growth. So to answer this question, we use the sphere drop model. In this model, use the two critical depth is mixed layer depth and critical depth. The depth where total photosynthesis equal total respiration. Below the depth, the phytoplankton cannot get enough sunlight to grow. So in the model, when the mixed layer depth shallower than the critical depth, the phytoplankton can grow. So it have higher chlorophyll. So in our study, we can see this is the critical depth on every day. So it's one day is have one peak. And this shows the mixed layer depth. And this shows the mixed layer depth was generally shallower than critical depth. This suggests that limit was not a limit factor for growth. So we want to know phytoplankton respond to sunlight immediately or they need time to accumulate energy. So we calculate the average sunlight over different periods and use coloration analysis to compare it with the chlorophyll. And the Y is the coloration coefficient. The X is average time. The picture shows chlorophyll is highly related to about 14 hours of accumulated light. And its coloration coefficient is close to 1. Next, we use the 14-hour accumulated sunlight to calculate. And here is the critical depth, and the orange is mass concentration of the chlorophyll. It shows the critical and chlorophyll follow a very similar daily pattern. This suggests that chlorophyll need time to respond to light. Finally, solar radiation is the primary driver of Diel variation in the ocean. Temperature, chlorophyll, and dissolved oxygen are response change in the sunlight. The decrease in surface chlorophyll during noon is likely due to photoacclimation and vertical migration. And the study also highlights the chlorophyll is not an immediate response to sunlight, but rather reflect accumulated light over time. Thank you.